This is CG Cancer Genome Workbench demo for this user. So to access the URL, you just type in cgwb.nci.nih.gov. And once you get to that, what you need to do is notice there's a couple of buttons on top. One is a tutorial that if you click, it will give you information about a comprehensive tutorial that you can all examples. And data dump, basically dump out all the mutations that were stored from different projects. And if you are a certified user, you could always use the login and type in your username and password to access restricted data that require data approval process. So over here is just an example of how to log in. So currently, the Cancer Genome Workbench have 5 different projects: the Cancer Genome Atlas project, Target, Tumor Sequencing Project, Cosmic, Hopkins Pancreatic Cancer, and the GBM, GSK cell line information, and Rembrandt copy number data. And we are also supporting individual small laboratories who are interested in collaborating with us, and they will send their cancer genomic data to us, and we can upload these and put them on the web page. There's a couple of different ways for navigation. The first one, easiest, is just typing a chromosome region or a gene of interest here, and you can just follow the lead, go where you want. You could also, so I'll just do a check, shows TP53. You could also limit the search to a specific using a tissue browser. Over here, say if I limit the search to brain as well as central nervous system, what you ended up is only observing the projects that have brain tissue or also central nervous system. As you can see here, not all the projects will be seen. And in addition to that, you could always select a particular project, like the Cancer Genome Atlas project, and pick a gene of interest. So there's a sub-project underneath the super project TCG that's showing up. There's the GBM data coming from various sources, from different sequencing centers, validated data, as well as ovarian cancer project of TCGA. So to show something most recent, I'll pick on the next generation sequencing data generated for the TCGA ovarian cancer. And the technology that we're using the exon capture. So we can see that once the project is selected, the genes that were sorted based on the those genes with the highest mutation frequency to the lowest. So for the ovarian cancer, we are seeing that P53 actually have the highest mutation, 63%, based on the next generation sequencing data. So what you are seeing here in the next window, once you select this gene, is a view that displays both the identified somatic mutations as well as cover each basis for this for each sample. And there's different ways of viewing coverage. This view basically scales these coverage by log scale. So the first line is 4x coverage, 16, 64, and 256. The red lines indicating the coverage for the tumor samples, and the blue lines is for the normal samples. So in cases when there's a copy number variations, you could actually see if there's any distance between the tumor and normal, indicating whether there's amplification or deletion that happening in the tumor sample. And this vertical bars indicating a somatic mutation. The colors over here indicating whether it's a missense, silent, or nonsense frame shift mutations, and the little bar here we're indicating whether this is a somatic or germline mutation. So since germline variants are only accessible to people who have logged in, and some of the bars you're seeing the black and colors split in half, and the black top indicating the proportions of wild type alleles in the tumor sample, and the colored one are indicating the mutant allele. Mouse over, you get more detailed information about each of these mutations, what amino acid changes the causes. And To get detailed information about the color map, you could always go here and find out what the color means for SNP classification, as well as whether SNPs, the disease status of each variant. And if you're a user who have logged in, you actually can view the mutant, the next-gen sequencing data, looking at the alignment view generated from the BAM files of next-generation sequencing data. And I'll use an example by clicking over here. And don't worry, you can always trust this. And it will create a picture that centers around where the mutation allele is. It may be difficult to view this with all the reads around. So one option this viewer does give to you is you could only display reads aligned at this site. And now we can see that, you know, in the tumor, in the normal sample, which is displayed in blue color here, there's no variant. Everything is consistent with the reference read. But in tumor reads, where we start to see the minor allele, the A to G differences among these at this site. And to be more specific, you could also see only show non-reference reads, that option here. And we can see that all of the tumor reads appear to have, a lot of tumor reads have the non-reference alleles in both orientations at the specific site. And you can also view, again, amino acid changes by clicking on this top button of showing check for amino acid changes. And another way of looking at this is that you also play the data in parallel with next-gen sequencing data to see if the mutation profile is consistent or not. So get to the data, you can go scroll down to the control panel over here and look into the mutations in denso, for example, for on that. And this is a view of showing somatic mutations from the Sanger data set. You actually can click on each of that. That will give you additional information. So over here shows the mutant alleles, flanking bases, as well as the amino acid changes it causes. And if you mouse over, you will see deleterious effect of these changes in the protein domain. And you could also view the traces that evaluate whether this somatic mutation is real or not real. So again, this is a view, launches the trace view, and people can see whether this is trustworthy mutation. And again, the A allele is very strong here. So in addition to this mode of looking at that, you could actually see in more detail whether percentage of variants, whether they have the percentage of tumors, whether they have a sample have tumor or not, in a more detailed view. Again, this full view that will show the identifiers of tumor samples and only visible to user who has logged in. And you can pre-select, deselect everything that's intronic or UTRs, and only allowed for display of somatic mutations, but not all the other types. And submit, it will give you a full view that you see whether how much samples by Sanger sequencing that you'll be able to observe. It almost look like all samples have somatic mutations in P53. So this full view actually says impaired tumor normal reads. The normal samples on the top and tumors bottom, and when there's a genotype discrepancies, it will show with a white bar. And again, there's annotations, their help document with details of everything. And we can see that some of consistent with what we found in the next generation sequencing data, most alleles appear to be homozygous in Sanger Center's sequencing data. And for some of these that were known somatic mutations, you could always go looking at links to Cosmic, like this one here, and it would take you to Cosmic so that you can see